Good morning, everyone. As Chris said, I'm Lynn Langit, and
I'm Denis Bauer.
So do you want to talk at all about your team?
Yes, so as I said, I'm Denis Bauer. I'm from CSIRO, which is Australia's government research agency. At CSIRO, we're passionate about bringing or translating research into products that people use in their everyday life, and probably our most famous product is Wi-Fi. There will be some speakers later today from Data 61, which is one business unit. My business unit is a different one. I am from health and biosecurity. So my team is working in health, human health. So therefore, we're developing technology, big data technology, in order to improve lives in general. So Lynn, how does big data look in your field?
Well, one of the reasons that we're working together is I'm a cloud architect, and I like to work with big data sets. And I was reading a study about the direction of big data, and you can see from this that in 2025, the sort of traditional data sources like YouTube and Twitter and astronomical data are going to grow into the petabyte size. But that's not really all that we have.
That's right. So talking about big data, nature is the ultimate hero in that space. So the human genome is 3 billion letters long, and it encodes everything from how cells look like, every tissue in your body is encoded in the genome, to how these things interact. So if it's a hot environment, if there's a famine, how to react to that. So when it comes to big data, the genome is probably the ultimate master there.
And in addition to that, you wanted to talk about genomics and human health and disease impacts.
That's right. So the genome does not only encode general things like the tissues, but also how we look like, how the risk of diseases, and how we perceive taste or our behavior. So an example there, it would be that the hitchhiker's thumb. So half of the people in the audience will have a thumb where the last digit extends further than the straight line. So I don't have a hitchhiker's thumb, but you can test. So half of you should have a more extendable thumb. Similarly with coriander. So there will be 10% in the audience that hate coriander. We're the coriander haters. So it's not your fault, it's actually your genome. So there is a mutation in the gene that changes the way that you can perceive taste. So you're actually a super taster for that. And apparently coriander has such a strong taste that you have an aversion to it. And of course, disease risks, so cystic fibrosis, everyone is familiar with that, which is a genetic disease that causes the lung to slowly fill up with fluid, which is encoded in the genome as well.
So genomics in 2025 will be the ultimate big data category because of the decreasing price of sequencing. There's prediction that between 25 and 50% of the entire world population will be sequenced, which is going to open up dramatic new possibilities in terms of human health and the quality of human health. And that's why I got interested in this field, and of course Dr. Bauer works in this field.
That's right. So we talked about how the differences in the human genome really come to play. So when you look at the person next to you, 99% of the genome will be the same, but there are on average 2,000 locations in the genome that are different, where you do have a different base than the person next to you. The person next to you. So from that perspective, this is how the data looks like. So you do have the chromosome listed, so we have 23 chromosomes, and the positions, which are these long strings of letters, because ultimately there are 3 billion letters in the genome. And then going all the way to the other side, what we see there is a 0/0, which is basically the encoding of what letter you have at that position, whether it is reference. So this is, in 2000 there was the first human genome sequenced, and this is our reference. This is how we, say a human looks like. And if you have a difference to that location, then it will be encoded by saying, so for example, a 1 means that you have an alternative letter there. And since we have alleles from both our mother and our father, we do have 2 copies of those things. So therefore 0/0 means you're absolutely like that genome that was sequenced, and 0/1 means you have 1 difference at that location. So therefore, the workflow, when we talk about how to identify disease genes, is like this. So you first collect blood from individuals. You then detect the differences in the genome. So this will be the genomic profile that we just talked about. On average, 2 million differences between each individual. From there, you then compare the actual, or you collect a lot of probes, a lot of individuals, and compare their profiles in order to identify what is the difference between healthy individuals and the ones that have the disease. With ultimately the goal, you find a hypothesis of which gene would be responsible for the disease, and then you put that in an animal model, say, in order to verify and validate that this is actually the cause for the disease. And from there, obviously, it will be, the next natural thing will be to find a cure. And this is a slide that I poached from the Project MIND, which is an international consortium with the aim of curing ALS, which is a motor neuron disease that Stephen Hawking has, and you might be familiar from the Ice Bucket Challenge. So we are the Australian node, contributing 800 individuals to the 25,000 individuals that will be collected worldwide.
So to be clear, the work that we're talking about today is already being used in research around disease and genomics.
That's right. So therefore, the focus of my team is to collect individuals and really find what is the difference in these large cohorts. And this is basically like finding the needle in the haystack, 3 billion letters again. Finding the single mutation that is causative is quite a daunting task. So therefore, just to conceptualize this a little bit further, we do have the genome. In the genome, there are certain genes, and we collect, and these are the differences between each individual, so 2 million on average. Then we collect the healthy and the disease individuals. So in ALS, for example, the people with ALS and normal healthy individuals. And then we want to identify what is the causative mutation. So these will be, for example, here, all the mutations that are present in the diseased ones but absent in the healthy ones. Now, obviously, this is an oversimplification, but for the purpose of this, that's the way that I think about it. So therefore, for the machine learning people in the audience, this is a classical machine learning problem, right? Where you have your samples and you have your features, and you want to know which feature is associated with the label being healthy or disease.
So when I started collaborating with Dr. Bauer's team, they were working with an on-premise Hadoop cluster, and one of the aspects of their work is they want to be able to share it with bioinformaticians worldwide. And so given my cloud expertise, I met with them, and I've been working with them around sort of the classic way that you build out a big data pipeline in cloud, which some of those steps they were familiar with, but some of the steps, because they are a research and a production organization, were relatively new to them. So again, this is just sort of more generic for big data problems in general, but it applies to bioinformatics too. We've clearly defined the business problem here. We're looking for the needle in the haystack. We have shown you what the data looks like. It's the VCF files coming off the sequencers. And then where we're really working is in the areas of candidate technologies around analysis, because it's computationally quite extensive and could be expensive, also slow. And so we're trying to use cloud best practices so that her team can get faster feedback, because as you're looking for these needles in the haystack, the faster you can go, the faster you can do tests, the faster you can get potential solutions out into the world. And in fact, the research is really interesting in that some of the applications of this, some of you might have heard of the CRISPR technology, we're going to talk about that a little bit more coming up, is being approved in more and more countries for applications in human health. So time really does matter, time and costs. So we're in the kind of middle of this where we're trying out different candidate technologies, and we wanted to share this talk with the big data community because these kind of lessons are applicable not only for bioinformatics, but for other types of big data like IoT, where the scales of data are such that we haven't seen before. So for me as a cloud architect, it's really interesting to work in this space because this is the ultimate big data problem, and also obviously it has profound potential implications on human health. So another aspect of this is machine learning at scale. So one of the situations that I find in this pattern is that you have this aspect of visualizations throughout the process. And again, I think I have a pretty heavy data science audience in here, so you're probably pretty familiar with concepts like Jupyter Notebooks and having visualizations as you do your experiments. But coming from the enterprise side, these type of techniques are very new to processing enterprise data. So in addition to helping Dr. Bauer's team, I'm trying to create reference use cases that can help with big data more generally. So your solution when you started was?
Yeah, so we originally came from a high-performance compute background. So everything we did was in a high-performance compute framework of multi-threading and then trying to identify independent tasks. Now, this is not the case anymore when you do machine learning on big data, because ultimately you want to use the whole data set in order to infer differences. Therefore, we did have to go to, it was our pleasure to actually go to Hadoop, because there we had these paradigms of parallelization that we can just harness. We did not have to implement our own OpenMPI workflow, but we can just use formalized paradigms. And this is really important for bioinformatics because bioinformatics, the analyses are quite short-lived. So there's constant research, which means that if there's one best practice today, it will be outdated tomorrow. Therefore, you need to adapt to that. And using Hadoop and its standardized framework is a much more agile way of doing things. The only problem with Hadoop, you need to go back.
Okay.
The only problem with Hadoop was that, as you know, the standard Hadoop used to be very rigid, and there was everything you had to be written to the drive. And again, this is not really amenable to machine learning. Therefore, Spark was the clear next step in order to do that. So what we developed is we created domain-specific machine learning library to deal with bioinformatics questions or specifically genomic questions. And therefore we can utilize the cloud pipeline patterns, which again comes back to that is standardized way of really being agile to create workflows quite quickly. Sorry.
Okay, well we pretty much skip through this anyway. So they started with an on-premise cluster with Hadoop, and then they have a library which is available in GitHub if you guys want to take a look at it, which we want you to take a look at it. It's one of the reasons that we're doing this talk, because it is out there for public review and use possibly in scenarios other than bioinformatics. So now, Spark. Why Spark?
Okay, so again, Spark was sort of the natural solution for the machine learning space, where we want to keep data in memory in order to come back to the node and reiterate over it in order to learn from it. The other obvious reason for going to Spark was that it's faster than Hadoop because of those in-memory computations.
So just a question, there are other in-memory frameworks out there, such as Storm and some other ones. Did you have time to evaluate other frameworks, or did you just go with Spark? What was your kind of thought process around that?
Yeah, so basically when we started back then, Spark was the only thing that was available, and we did not test anything in the meantime, because Spark works so well for us in this particular paradigm. All right, so you wrote VariantSpark, and it's been an interesting evolution of this custom machine learning API. When you first started, you started by writing on top of Spark machine learning, or Spark ML, and you did an implementation of k-means clustering or grouping. But that didn't work very well ultimately. Why? Well, it worked well for the data set that we had at hand, which was the 1,000 Genomes project, so there will be 2,000 individuals and 80 million variants. And k-means clustering was fine because it's not a iterative machine learning question. It's that you want to identify the means and optimize over that. So this is easily parallelizable, easily somewhat independent. Therefore, when we went to and our application case was to predict the ethnicity of individuals, or the ethnicity of these 2,000 individuals, and it worked reasonably well. Problem though is that k-means clustering obviously is a very crude approach, and it would be nice to train a more sophisticated method, specifically since we have the labels. So therefore we wanted to go to a supervised machine learning framework. And there Spark really fell down. The Spark ML lib just could not handle the amount of the length of the feature set that we are dealing with. And the reason for that really is that it was designed for the big data in Lynn's world, which is lots and lots of samples, and the description per sample is quite minimal. So these will be like the things that you're familiar with, the age of people, where they live, what the sentiment is, and things like that. So usually it's human annotated or human collected information that is 100 features. And even when you go out to automatically selected features, you would not get more than 10,000 of it. But in our case, this is 2 orders of magnitude too small for our problem, because we have millions of features. Therefore, we had to come up with a different way of approaching this.
So you guys are all data scientists. Did you hear what she just said? This method of analyzing accounts for how many million features?
80 million in that case.
Did you say it again? How many million features?
80 million.
She's kind of modest. She's kind of modest. So 80 million features. So it's a very interesting implementation, and really this is kind of what compelled me to get engaged with her group. When I came out and I was visiting, she said, yeah, we have this library that we've written and we published on it and we're interested in putting it on the cloud. And I said, explain it to me. And she said, million, and I made her say it a couple of times, 80 million features. That's really interesting, you guys. It has a really broad applicability in the world of big data that we're coming into, like I said, IoT and some of these other domains, regardless of bioinformatics. So the implementation is random forest with decision trees. So I'm sure a lot of you guys would be familiar, and the magic is the splits. You're splitting this way and this way, yeah?
Yeah, that's right.
Yeah, there you go.
So the traditional implementation of the Planet implementation from Google was to split features by samples. Yeah, so splitting features by samples. But the problem there is that you cannot fit that many variant or features into the memory of the nodes. Therefore, Yahoo and Berkeley, I think it was Berkeley, came up with a different way of splitting it, namely to split it the other way around, to fan out samples rather than features and collect them back together. And that worked very well in terms of 10,000 of samples, as I said, the automatically collected features, but again it wasn't applicable to us. So therefore we had to go one step further in order to split it or make it more independent, and therefore our smallest operating nucleus is basically the cell. So it will be one sample, one feature, and have that to fan out and collect back together as you create the subsets to build random forest. So I don't know how familiar you are with random forest in general, but random forests are basically a set of or an ensemble of decision trees. Therefore, as a decision tree, it tries to split the samples based on their label, and it splits it on the features. So therefore, when you have a couple of hierarchies, you have split in my case, it will be you splitting on gene X, splitting on gene Y, and splitting on gene Z, and therefore you separate out your data. And we created a methodology that would do that simultaneously over all trees, over all or nearly all possibilities that you have there. So with that, it's and. Person who just walked in is actually the architect of this. So what we have here is basically the middle bit here, where we identify the best split over all possible combinations that we look at. And therefore, this is radically different to what your traditional vanilla random forest is like, because now we can nearly exhaustively sample the whole space by just building that many brute force number of trees and evaluating the actual splits that we use. So therefore, the application case will be here classification that I'm sure you're familiar with, but also identifying which feature are associated with the label that we call association testing in the genetic space, but you probably call feature selection.
So it's one thing to see a GitHub repo, but we're talking about important, life-changing work. So, you'd think maybe we're going to show you some cancer genomic sample. We can't do that because of the privacy of data. So we looked at a different disease condition, and that disease condition is hipsterdom. Now, we had to talk to some people in Melbourne because, we're in Australia. And so we have a demonstration that we're going to show you with a synthetic phenotype, which is her words for fake disease. So let me switch over and do that. So inside of here, we are using the Databricks Spark as a service platform. And one of the reasons that we made this as our demo is because it's very reproducible, and we really want more people to be able to try this out. We really want people to be able to just quickly set this up. So you can get for free a Databricks Spark cluster for 2 hours, just for demonstration. And it's a Jupyter-style notebook, so you just click Create Cluster. And then we have instructions inside of here where you can associate this with the VariantSpark library, which we've published to a Maven repository, so that we have a working version, basically. And we have the instructions on how you do that. And then what we've done is we've created a sample notebook, and we'll give you the link at the end of the talk today, so you guys can party on the data. It'd be really fun. You can use this data, or you can use other genomic data, or really any data to see how the algorithm works. So I'm going to let Dr. Bauer explain about this hipster disease section, so this section of the notebook.
Yes, so in order to run really with the paradigm, what we want to do is create a synthetic data set that would mimic how complex diseases actually work. So therefore, to make it fun, it was the HipsterIndex, and then to run with that, we actually picked locations in the genome. We could have picked any location in the genome, or we picked locations in the genome that are known to be associated with traits that we would describe as hipster, the elements of hipster, of having a monobrow, having facial hairs. So therefore, I think the first one on there is on chromosome 2, there's a SNP that is associated with monobrow, and it was published in the 2016 Nature Communications paper. Similarly with the retina horizontal cells. So this is a cell type in your retina that can detect color differences or shade differences. So therefore, we're inferring that if you have that, then you might prefer checked shirts because there are so many color differences in there.
I'm looking for the hipsters in the audience, but anyway.
I can see a couple, for sure. Similarly with the facial hair and the coffee consumption. So there is actually a SNP that makes you metabolize caffeine more quickly. Therefore, people that have that mutation have the tendency of drinking more coffee. So therefore, we took all of those locations that are known to be associated with the phenotype that we would describe as a hipster, and then created a score out of it that mimics a complex disease. So these will be additive effects as well as multiplicative effects, as well as that some of those contributions are weighted. So for example, the top one that we're weighting is the monobrow. So we say every hipster has a monobrow. And down here, Lynn, when you point to the actual formula, this is the formula that we calculate. So we take for the 1,000 Genomes project, we take the actual genotype of that individual, or those 4 genotypes of the individual, and calculate this score for every individual in the 1,000 Genomes project. And if that score is above 10, then it is a hipster. If it's below 0, then that person is fine, he's not a hipster. And we annotated the whole genome with these 0 and 1 labels.
So this is the disease, and then the idea is they're testing their algorithm. So it's kind of like a test-driven development because they're labeling the data, running the analysis, and then they're expecting the results to match. So it's a proof of concept. So again, I'm assuming that most of this audience has seen a Jupyter notebook. This is a Databricks notebook, which is proprietary format, but similar conceptually in that this is like a wiki. But it allows you to run also computational blocks. So you have Scala here as the default language. So this is a new method of working that I assume many of you use, but in my world, in the enterprise, a lot of times when I show developers, they've never seen this type of thing before, which might be an interesting perspective for you. So this allows you to have annotation around your experiment. So this is the setup, which is inside the notebook, and this is publicly published. So again, you can try this out, and we hope you do. So this tells you the steps to set up the cluster. And then once you've got the cluster, then you need to load your data. And one of the great things inside of these notebooks, of course, is you have the different runtimes as long as they're loaded into the cluster, and Databricks has the ability to use Python. So these are executable, so you just run this, and then this is going to be loading up your sample data, and notice it's of type VCF. And then once you load your sample data, then you're going to go ahead and call this algorithm that her team has written, this API. So instead of a Spark context, you're going to use a VS context. You're going to wrap that around the Spark context. And then you're going to have your feature sources, and this is basically your IDs, your first 10 IDs. And again, for purposes of time, I'm just going to kind of run down. And now we're going to label. And this is where we label based on the characteristics of the samples, whether they're got the hipster disease or not. Does that make sense? And then we run that. And then we can run our analysis. Now this is really the key aspect of working with the algorithm. Dr. Bauer and her team, and Piotr, who's here, worked really hard to wrap the random forest to make it simpler to use, fewer lines of code, because what is the goal of the bioinformaticians? To run these samples through without writing a lot of code and get their feedback, so they can find out where the significant variances are and then continue their experiments. So you create an instance of the API, just passing in the feature source, the label source, and the number of trees, and then you run the analysis. So it's really just 4 lines of code to run analysis. And this is quite different, obviously, than writing a custom algorithm, or even in some of the other genomics libraries that are out there. So once you run the analysis, then you see the results, and you can remember that the monobrow was the most important. And you can see coming out, here are the results. And this was computationally intensive. We ran 1,000 trees, and it took a great 3 minutes, which is one of the reasons you use the cloud versus an other type of solution. Another thing that's interesting about Databricks is inside of here, you have integration with all of your Spark logs. So what you can see is you can see the overhead of the execution, and you can then iterate in a production environment, and you can buy more Databricks units, because basically Spark as a service. Now, I don't work for Databricks, but we went for the software as a service because, again, from her world, she doesn't want to have a lot of administrative overhead, Hadoop admins, that kind of stuff. She doesn't want to wait for compute time. She just wants to put her data in, run her algorithm, know what her costs are, and get her results. So this integrated environment is interesting not only for demo, but possibly for actual use. We're actually exploring different alternatives as we go. So then, once you get the result, then one of the great things about the notebook, and this is alluded to what I talked about earlier, is you might want to visualize it. This talks a little bit more about the way that the impurity is calculated. So a lot of you guys are data scientists. I'll kind of just let you read that if you want to. You can see here when you visualize the results as a default, it comes out as a table. And this is actually using Spark SQL. Again, one of the neat things about this environment is you can use whatever analysis is comfortable. And another neat thing is you can visualize. So Databricks itself has these visualizers built in, and you can then work with them in a number of different ways. But that might not be enough for you because visualizations are important. So we've taken the same results and we visualized them using Python because we can. And because, we can angle this because that's what we get paid the big money for, so that we can see the results better, right? And we can even use R. I would assume most people in this room are very comfortable with R. So we've also demonstrated here in Spark 2.2, we're on the latest version of Spark, how you work with Spark R versus R, which is slightly different. It's changed in Spark, which can be annoying. So we've got an example of it here. And then we have a visualization where we just flipped so that we can see that the variants of interest here are 2, 5, 7, and 4 should be on there. So we're actually accurately predicting. And I'll let Dr. Bauer explain the results.
That's right.
Yeah.
So remember, this was a proof of principle or a concept that our algorithm is actually accurately finding what we put in the data. So just a recap again, what we've done is we take the 1,000 individuals, annotate them with 0 and 1, and basically this is all we gave VariantSpark. The full genome profile as well as a 0-1 label, expecting it to find the actual individual locations that contributed to the disease. And we were very fortunate and pleased that it actually find exactly those locations that we put in.
So
and it did not only find the exact locations, but also by a quite large margin. So when you see it down here, we have the chromosome listed, so this is the whole chromosome. And on the y-axis, we do have the impact that particular feature, and each dot is a feature, is a SNP. And on the y-axis is the actual contribution of that SNP to the disease, or the association to the disease. And what you can see is that the 4 locations that we put in by wide margin have the strongest association to the actual disease. So therefore, if we put in a real disease in there, we would expect it to find the genomic locations that contribute to the disease. And it's not only the single locations independently, but also the interactions of those.
And continuing on, I won't take too much more time on this because you can look at it if you're interested, but we brought in an alternative framework called HAIL. We did an analysis on the same data. We then brought it in, and we brought it into R, and then I'll just make it smaller so you can see. And it was really interesting and really obvious and really important that the salmon-colored dots are the results of VariantSpark, and they're different.
Yes, exactly. So HAIL is a different underlying methodology. One could argue a simpler methodology because it's not machine learning as such. Therefore, it was clear that our methodology brings out the exact features and only these features in the right order, whereas what HAIL produced was it found those features but in a different order, and on top of that, which is the cloud down here, it found a whole suite of other things that clearly have nothing to do with the actual phenotype, because we know what has to do with the phenotype.
Exactly. So, all sort of smiling aside, this is profound, and this is why I get on a plane from America and take that 14-hour flight many times, because I feel what she and her team are doing is very, very important in bioinformatics and in other domains. So the way that this was built is VariantSpark with Spark on Databricks, but you could use any Spark implementation. So you could use Amazon EMR, you could use the Google version of it, you could use the Azure version of it. We just happen to use Databricks because it's quicker to spin up. And all these frameworks are installed, and then we use S3, and we use an Amazon cluster, and then we have an associated Databricks notebook in the demo. You could also do this with a Jupyter notebook. And you got some good results with performance when you started working with this internally, right?
Yeah, that's right. So VariantSpark was the only methodology that we tested that could scale to the full dataset. And scaling to the full dataset brings a couple of perks with it. So one is accuracy, obviously. Other methodologies could not take the whole dataset. They only could take subsets of those. And while the results were, they're good proxies, they were not as accurate as taking the full dataset into account. Similarly, with the speed, our methodology was the only thing that that was quick enough and scalable enough to deal with the workload that we threw at it. So, and this is to show you that the dataset that we took, scaling it up is not a problem. So what I have on the x-axis is the number of features. So these would be the whole genome or a subset of the genome. And what you can see is sort of it's scaling linearly. As we throw in more features, it scales linearly. But it also scales linearly with the number of samples that we throw in. And this is not surprising because this is Spark, this is what it's designed for, but this is a nice validation that that's the case. And actually it's sublinear. So from 1,000 to 5,000 is a larger gap than from 5,000 to 10,000. And so with this, I only want to show you that it's scaling with a number of features as well as with a number of samples.
So in addition to this, we're currently in the process of performance testing. Databricks claims that their version of Spark is 30% faster than any other version because Databricks is the company where the Spark committers live and work. And so you pay more because it's software as a service. So as we proceed with this work, we intend to publish it, and I think this is going to be very interesting for a lot of people in a lot of domains. So we're going to be comparing Amazon EMR performance at high scale with Databricks at high scale. So this is work that as a cloud big data person, I'm really excited to see the results and to work with Denis and her team to publish. So you can try it out, and these slides will be made available. We published on the Databricks blog that notebook, and like I said, Databricks offers in the community edition a single node cluster for 2 hours, where the cluster is ephemeral and the notebook will persist. Also, if you are a researcher, Databricks has an academic program, and we're working on it now, where you get a 6-node cluster permanently, so you can try it for free. So go contact me, and I can connect you to the Databricks people if you want to try out at a little higher scale. So now we're going to switch to something different. Oh no, we're going to talk about one more thing.
First, we talk about what is our roadmap.
Yeah.
You know, what we want to do next, because this technology currently is really geared towards genomics, because it was the first discipline that has that problem of large features. But with the datification of everything, basically this problem will come to all and any other discipline. Therefore, what we want to make VariantSpark available, or the features that we want to put in there, is to cater for those other areas. So specifically, we want to put in the ability of joining features that have mixed properties, so continuous variable with categorical variables. So in my space, there will be genomics data joined together with electronic health records, for example. And also we want to increase the ability of dealing with continuous variable of different value ranges. So again, in my case, there will be protein expression data, for example, where one can go from 0 to 100 and the other one goes from 0 to 1,000 or 10,000, and therefore making the random forest methodology agnostic to the actual differences in the value ranges.
Right.
And I'm sure you can think of examples in your case where this will be really helpful.
All right, now we're going to go into something else. So what's this?
That's right. So now that we, in the audience, now that we have identified the actual disease-causing mutation, hipster, there's this new technology, a fundamental shift in the medical space, that we're now able to edit genomes of living organisms in order to change the trait of that organism. And specifically, the application case here is that this has the potential of eradicating genetic diseases. So this has really the potential of bringing in a golden age of medical science. For example, how close we actually are was demonstrated by a publication just a month ago in Nature, which edited human embryos to remove a genetic disease called hypertrophic cardiomyopathy, which is a heart disease. So 1 in 500 Australians are actually suffering from that heart disease, which causes the heart wall to thicken, and the worst case outcome is a sudden cardiac arrest. And some people do not even know that they're carrying this genetic disease. So it's a monogenic disease that you can just edit out. And this is what the paper has actually done. The problem, though, is that it only works in 7 out of 10 embryos. So when you think of this being your unborn child, then 3 out of 10 failure rate is just not good enough. Therefore, what we're doing, or what we're trying to achieve, is to build a computational methodology which aims to improve this success rate to make it work the first time, every time. So obviously, being software engineers, the first thing that we've done is looked at to making it quicker. And the reason for that is that TT-scan, basically you can think of it as the search engine for the genome. So researchers can type in which gene they want to target and how they want to target it, and then it comes up with a ranked list of possible options of how to do it, so researchers know straight away what is the best choice. Therefore, if you want to do that, you probably want to query a lot of different methodology or different ways of interacting or targeting a certain gene. Therefore, making it quicker allows you to interrogate different parameter sets and find the actual optimal solution faster. So therefore, what we've done is we parallelized out the search, and it therefore reduced it to actual seconds for each gene. But researchers might not want to just search one gene, but they want to search, say, 100,000 genes. Therefore, making it scalable, or something that just takes a second to execute, is quite tricky, and it was only recently been made possible in the cloud by something that is called the serverless architecture, so Lambda function in AWS. Therefore, we implemented a serverless architecture harnessing Lambda function in order to really parallelize it out and collect it back together, the information of things that only take a second to execute.
So this is actually how I met Dr. Bauer. I was sitting on my couch in California, and I was reading the Amazon blog from Jeff Barr, who's the lead evangelist, and I saw this architecture, which is her architecture, her and her team, and it was commended in his blog as outstanding serverless architecture being applied to a very important problem. And I said, who is this person? I have to meet this person because my enterprise customers can't even figure this out. Who's this bioinformatician? So I literally just reached out to Dr. Bauer on LinkedIn and I said, I'm going to happen to be in Australia in February and I would like to come over and talk to you. So of course she's this very smart person and she looked me up and saw that I was an Amazon community hero and said, you can come and talk to me, but I would also like you to present on all the new Amazon technologies so that you make good use of my time.
I did that, didn't I?
Yeah, she made me work for my visit. So of course I then got on that and did that. I think it's just a really fantastic example of the application of modern technologies in sort of unexpected places. Most of the time when I've talked with bioinformaticians, they're working on local clusters or they're working on EC2 instances, and the fact that she is able to bring together a team that can implement this after going to an AWS event and seeing the architecture is really kudos to her, and she and her team are getting lots and lots of justified awards. You're a very modest person, but it's just fantastic. She's gonna you did an interview and you're going to be on This Is My Architecture about this, and you're of course always looking to optimize this architecture. Now before we talk about the optimization, we should actually show the service, so let's actually show it and you can kind of guide me through. This is the service running live, so tell me what to do here.
Okay, so remember this is the search engine for the genome where researchers would want to click and find the optimal target site in order to edit the gene. So therefore if you go to the submission page.
Okay, so scan?
Yes.
Okay.
So in this submission page, people can submit the actual genomic location they want to query. So Lynn, the shortcut for you is to just load an experiment that we have put up already. Yeah. So this is a genomic location. It has a start and stop location, and it automatically fetches the actual sequence that is underlying it because it knows what the reference genomes look like. So therefore, when you submit that, it will start several Lambda functions. So the first one of those Lambda functions will actually scan this genomic locations for all the possible target sites in there. So this is just a simple regular expression that finds strings of a certain ending. And this is basically what we're seeing here is all these black bars are possible target sites. So this could be what a researcher is designing the technology to do to target. So as you can see, even in this small example, there are lots and lots of choices that people can make. And typically people, I wouldn't say random, but it's sort of almost random the way that people usually pick their target site. Now, our solution is not random. What we're doing is we're evaluating all these target sites with respect to their ability to actually attract the genetic machinery. So the genetic machinery is binding to the DNA and doing something there in order to make the edit over there. But it could bind to other locations in the genome. So therefore, since you have a string of 3 billion letters, having the occurrence of one motif can be random, because these are just 25 base pair long. And when you think of the combinatorics of 3 billion times 25, and the genome only consists of 4 letters, you have lots of opportunities of this particular sequence that you found over here to occur somewhere else in a gene that you certainly do not want to edit. So therefore, what we're doing in the background here is searching the full genome for every one of these locations. And this individually is a Lambda function as well. So therefore, you might have noticed that it takes a while to load up data. It's because it's spanning out Lambda function and collecting back the information on the fly. And as they become available, it's updating the front end here in order to annotate these sites. So therefore, all the green ones out here are good sites, are sites that have few off targets and good probability of resulting in a successful edit.
So I'm working on learning all this bioinformatics stuff. green is good, black is bad, okay? And we are running past time, so we're going to have to get to the end here, which is Dr. Bauer and her team have come up with these really innovative solutions around big data and bioinformatics that not only can revolutionize that domain, but other domains. And that's why we're really, really proud and happy to be able to share the work with all of you here. So the first problem is solved with Spark-based machine learning, and the second problem is solved with a serverless implementation. And this is implemented as VariantSpark and GTScan. And we really have to be respectful of time, so I think we have to finish. So thank you so much for listening in.
Thank you.